Recording in progress.
All right. Well, good morning, everyone, and welcome. Thank you for joining the November Going Organic seminar series today. My name is Tristan Lawrence, and I'm the program manager for the Pulse Quality and Nutritional Breeding Program at Clemson University. This seminar series aims to educate growers, researchers, and consumers on organic agriculture. Before we start, I would like to thank our sponsors, which is the U.S.A. National Institute of Food and Agriculture, U.S.A. Feed the Future, South Carolina Department of Agriculture, the Good Food Institute, and USDA Agriculture Research Services. So today we have Dr. Matthew Blair from Tennessee State University, who is a plant breeder and molecular geneticist. He specializes in practical and molecular marker-oriented plant breeding with experience in amaranth, common bean, maize, melons, potatoes, rice, sorghum, and soybeans. He's also a molecular geneticist with experience in gene cloning, marker development, QTL analysis, association mapping, and molecular breeding. He is the principal investigator for nutritional improvement in bean genomics and biofortification challenge program and Harvest Plus, Canada Linkage Fund, and Generation Challenge program grants. He also works with the Bill and Melinda Gates Foundation on abiotic stress tolerance breeding and the BMZ in Germany. At this time, I'm going to go hand it over to Dr. Blair, and we're excited to hear your talk.
Great. Hello. Let me mute you. Oops. How's that?
Sounds good.
Okay. Sorry that the video's not working. So if you could just miniaturize my face, because it's not going to change during the whole talk.
Your PowerPoint is not
everybody. And Tristan, thank you very much for that introduction. That was awesome. Especially the Dr. Bean logo, which I know you got from your advisor. And I'm sad that she's traveling right now, but Dill is a great friend of mine, and I hope she can join or listen to this under recording, and that some other professors that I know at Clemson can attend. And I've invited some of my students from Tennessee State, but I'll need to forward them the new Zoom because of the way it's linked, but they might already have joined. I see there's a good 30 participants. Oh, and even Yassine Torsh, a student from Algeria. That's great. Okay, so I will start with sharing my screen. And if you could confirm that that is visible.
Yep. Looks
okay?
Good. Yep, just enter presentation now. Perfect.
So the title of my talk is The Development of Mung bean as a nutritional quality product, and this is going to concentrate on pre-breeding assessment and DNA marker identification. So a little bit the items that Tristan touched on in my CV, that is where we're going. That's the focus of the lab. I'll introduce our lab, our group, and some of what we're doing. And it's a pleasure to be with Clemson University today. I visited Clemson, I want to say. And I really like the similarities between our two states. Tennessee and South Carolina share a little tiny bit of border. A little factoid for people: Tennessee is the state with the most number of neighbors. So we're just neighborly. So y'all are welcome to visit Nashville anytime you want. I know that's a great invitation. Okay. I'm going to give our present research focus in terms of the crops that we study. We work mainly on new crops, and so that includes pseudo cereals like the amaranth that Tristan mentioned. And then we also do some subtropical legumes as summer crops, and really have realized that our summer heat is an advantage, not always a disadvantage. And especially for mung bean, that wouldn't grow way up north, but it certainly grows well here. And as we face climate change, we have to have mitigation strategies. And one of those strategies is to have new crops. Even if they're not traditional for the US, they come from other parts of the world, like Southeast Asia and South Asia. And we can really do great things with those crops. And I think people at Clemson know more about this than anyone because of the wide variety of climates in the state, and because of Dill's work on new legumes for the winter season. Winters are getting hotter, which is an opportunity, just as summers are getting hotter. And when summers get hotter, we have to deal with drought and heat tolerance. Drought is mainly intermittent in Tennessee. We get enough rainfall, but it's just not distributed well in a lot of cases. And then we have emphasis on training of students, and we've had visiting researchers from a range of countries. For U.S.A. students, we have an emphasis on African American students, as we are a historically Black college. We're one of the 1890 universities, and we're the major 1890 for the state of Tennessee, which is also land grant. Just so that you don't confuse us with University of Tennessee, which is over in Knoxville. We're in Nashville, which is the capital of the state. I run a genomics and genetics laboratory, and we genotype with various markers. We use single nucleotide polymorphisms. We have a pipeline similar to what breeding companies use that I'll get into later. It's called CASP. Then we have genotyping by sequencing, which is obviously by its name, a sequencing-based method, and we work with mainly Illumina platforms for that. And then we're doing some marker conversion from GBS and association mapping with both of those techniques, but mainly GBS, and then training of students, as I mentioned. Oops. There we go. We're blessed to be in a new building. It's called Agricultural Biotechnology. It's based in the middle of a smaller ag campus, which is mainly new buildings. You can see what we call the barn to one side, and our tower has a DNA helix in it. So if you look really carefully at the lower picture, you'll see a DNA helix. Now, it was done by architects, not by scientists. So they didn't realize there were major and minor grooves to DNA, but at least they got two interweaving strands. I keep doing page down. There we go. The laboratory conditions are typical of a plant genomics lab. We use some robotics, like I say, to introduce students to what they'll see at private companies. And then we have benchtops for DNA manipulation and tissue culture rooms separate. You'll see some of the international students here, including Borlaug Fellows. The outline of my talk is to discuss mung beans as a new crop. Actually, I'll sort of switch this and discuss the Tennessee implications at the end with a varietal selection we've made, and then also comparing it to varieties that are already certified seed in other states. I'll introduce the crop. I'll give introduction to the USDA collection, which is where we really see the diversity that we can tap into here in the US. We have genetic marker development, a diversity study to get an idea of what gene pools are in the crop, and then GBS evaluation, which is tied into a mineral evaluation, which is part of the biofortification work that Tristan mentioned. So our first experiences with this crop was we actually started on amaranths, and that's what you see in the background. These are some of the vegetable types that are very leafy and prolific in hot summers here in Tennessee. And we needed a crop that we could multiply ourselves. And so we just went with some mung bean. We started with Seed Saver Exchange and other commercial sources. And one thing we noted right away was that it was a very uniform crop and grew very well. It was less expensive than soybean and faster in germinating. And so we started using it as a border in our amaranth fields to tell what our sort of diversity was and what our diversity of soils was, and also how our crop was growing. And you can see that the mung bean really did well. We also did an experience with some both GMO and non-GMO corn and sweet corn. And this is how mung bean is traditionally grown in China and East Asian countries. It's a intercrop, so it's very shade tolerant, and you can see that it grows very well. We actually found, as an aside, that it increased, we presume through nitrogen fixation or weed control or just the benefits of intercropping, 10% increase in sweet corn production. So it's just a little hint for people that want to grow something easy to harvest seed for and still get the benefits of a summer crop that they can just throw in their cornfield. And this was just side planted to corn and done with 50-inch row sweet corn. Okay. Everyone can still hear me, right?
Yes.
Okay, and see me. Unfortunately, yeah, the video's not working, but as long as you see the slides, you're good. So I come from a background of working with phaseolus dry beans, and mung bean is a vigna, just like cowpea. It's a new crop. It's heart healthy. It's got a lot of nutraceutical benefits. And here is a comparison of the three growing in Tennessee on our research farm in Nashville, along the Cumberland River downtown, basically near the downtown and near our campus. And among those three crops that you saw on the previous slide, dry bean is the earliest, and it's always ready in late July or August, and it really wimps out in the heat. So there's nothing you can do about it unless you want dry beans. Mung bean, on the other hand, will grow throughout the season and can be planted as a border to corn patches or bean patches. And just comparing it to other types of vignas, in this case, two types of black-eyed peas, the Crowders and the red clay, they are a little bit faster maturing, and they seem to be good in terms of nitrogen fixation. So we really started from a point of view of, okay, we're trying this exotic crop, mung bean, and we're going to see how it does. And this was back in 2015, but it really got me excited. And when we had visitors, this is a gentleman from a refugee, which Nashville has a lot of refugee population because it's a big city and people have been coming associated with churches to live in Nashville from all over the world. This guy is from Bhutan, and he's, I think, a person of Nepalese descent in Bhutan, and he's come to America because Bhutan closed its doors. And he is part of a larger group that grows different legumes, and they wanted to see some things, and they were fascinated by not only the mung beans, which they knew of, a little green bean, but they were also fascinated by the red clay peas and the Crowder peas, and both of those are sort of, you know, from your guys' neck of the woods over in South Carolina. Now, mung bean is really interesting because it has this long cultural history in South Asia, India, Bangladesh, Nepal. In Hindi, it's got all these different names. It even comes all the way from Sanskrit, and it's well known there. It's also well known in China, where it's called liu dou, and all the countries in between, from Bangladesh, Nepal, Philippines, Taiwan, all grow mung bean, and there's a collection in AVRDC or World Veg in Taiwan. It also grows in more desert climates like Pakistan and Iran and Afghanistan. So we found that interesting later on. As a bean, it fascinated me because it has special starch properties. It can be gelatinized and used to make jellies or noodles, and therefore it's useful as an ingredient, as a flour, and both as savory and sweet dishes. And so it's not like common bean that's just boiled and put in a can, as we often say, because we're one of the big canning states for beans. Its classification is, like I said, as a Vigna species, and it's basically a little green bean that's round. For those of you that don't know mung bean, it's the same bean used for bean sprouts. So some varieties with slightly larger seed are sprouted on wet paper, and they become your bean sprouts. So another little industry for Vigna radiata. There are 70 species in Vigna, and they're all from Africa and Asia. And there's quite a few domesticates in the Vigna genus. So from Bambara groundnut to cowpea to mung bean, rice bean, and black gram, which is actually the species name with the epithet mungo, is black gram. So we don't consider that part of the green gram, which is Vigna radiata. And there's a range in selfing and outcrossing capacity. There's surprising dishes, as I mentioned, sweet and savory. Chinese cakes made from mung, polenta, mixes with corn. It's one of the fastest cooking beans, so it actually boils at the same speed as rice, especially if it's decorticated. And it's common in dals. It could be used for making ice cream, but rice bean is preferred for that because it's got a nice red color. So that's a different big exclusively from Japan and China. But you do get the industrial uses of mung bean flour, like noodles and green bean starch. And the nutritional aspects are really good, that there's high level protein. It's complementary to cereals, especially for rice-based diets. I mentioned the mung bean sprouts, and it's also economically valuable, mainly in regional trade. But it would be interesting to have this be a major international commodity for us for trade from the U.S. The genome has been sequenced for mung bean. It shows that 64% of nuclear DNA is single copy sequences. So again, it's a small genome legume like common bean. Cowpea is relatively small. 46% of the single copy DNA is interspersed with repetitive sequences. As I said, the genome's small, the chromosomes are small, and they're difficult to study because of that. So the current estimate is that the genome is 579 megabases, or base pairs. We know that SNPs are the most common form of variation in any plant, but they're especially the most interesting source of variation for mung bean. I entered mung bean a little bit by doing simple sequence repeats and found that many of the simple sequence repeats are embedded in those gene regions, and so they don't have a lot of diversity compared to other legume species. And so really the SNPs are the way to go for mung bean, and also their ease of detection. So one other thing is that SNPs occur in haplotype blocks, so they're easy to associate haplotypes with certain alleles and with certain phenotypes in genome-wide association studies. So their marker benefits is they're the most recently developed DNA marker per se, and they're preferred in abundance, throughput, low-cost genotyping, and are applicable in a wide range of areas. The first part of the talk that I'll give is objectives that we had based on the genome having been sequenced in 2013. I arrived in Tennessee about that time, and we started with the full USDA core collection, but the part that we worked on was just basically a single 96-well plate of diversity to sort of start some testing. And we also started some testing with the CASP technique for SNPs. The USDA collection is basically around 400, 500 accessions. in a core collection and about a total of 4,000 accessions overall. And we planted the entire core collection as part of this work and obtained it from the Griffin, Georgia U.S.A. station. We planted over 2 years for our initial work, and the first was an unrandomized experiment with checks throughout the field, and it was planted in early June, which we determined was the best time for mung bean to be planted in Tennessee. It's when the soils have heated up a little bit, and also because they're drought and heat tolerant, that takes them through the summer, and then you harvest in September when they either dry off or in October when the first freeze comes. We did find there was an uneven harvest from early to late material from 3 to 5 months. And so we had to whittle down this collection of 400 genotypes based on trait measurements like grain yield and 100 seed weight, and also mineral content, which I'll describe in a second. And we had to whittle this down so you can see that we had interselected for our work with the first 140 that we used next year, and then 95 that were analyzed by DNA. So this is from the 400 collections, just showing the first group of accessions and how they originated from many countries around the world. So I'll indicate that, the accession name is PI for plant introduction, and then origin ranging from the U.S. to India and China being major providers, and some even from Iran, Thailand, Zimbabwe, so countries outside the sphere of mung bean. And I'm going to skip over some of these CASP assay slides because they're more for an audience specific to genetics. But CASP is an easy method to evaluate DNA polymorphisms because it has 3 PCR primers instead of 2 that are typical. And this is what you have as a master mix, and it has fluorescent dyes that makes it easy to detect on the following machines. You run a thermal cycling condition just like you would a PCR, but that polymerase chain reaction doesn't include as many steps of amplification as you would for a typical amplicon, because what you're trying to get is what's found here in the middle, a graph that separates the alleles into every one that contains homozygous GG and another group that has a homozygous AA, and those that are in the middle that have both alleles that are heterozygous. And we have all this equipment in our lab: the Meridian for robotic dispensing, the Cube for plate sealing, and the hydrocycler for PCR. And in our first work, we looked at 42 amplicons, all from gene sequences, and analyzed them for the single SNP that was determined to be in the middle of them. And these 11 were eliminated for multiple substitutions, and 33 were continued to use for CASP primer design. And all of these were good markers and gave these type of typical results. Again, big Y, big X, and XY as a heterozygous. And the good thing is you always have an internal control under this SNP fluorescent pattern, where you have the green dye on the left axis, and on the bottom axis you have the red dye. And you can really tell when there's a water or a non-template control down at the bottom that doesn't amplify and doesn't produce a signal. So we looked within Legume Information System to blast the genes that we were targeting and report on what those gene families were, and what their ontology was, and what the biological processes associated with each gene were. And I'm emphasizing these 33 genes just for 2 reasons. One is because they're the first ones that are used sort of as a common marker set. Surprisingly, There hasn't been much of that published in Asia. And secondly, because one of these genes turned out to be very interesting and associated with a wider study we did with GBS, where we found allele differences for iron accumulation, which is a nutritional trait in beans. And we wanted to follow up on that more in these subsequent studies. So these show the successful markers. I know it's hard to read, so I won't dwell on it. We just developed a naming system that you can see to the left. And that's the primer names, MB for mung bean and K for CASP, and then the SNP order series that we had, and then what chromosome it was on and what molecular and biological functions they had. Most SNPs were co-dominant, in fact, almost all. And you can see, again, a picture of the scan for a marker. A close-up of the MB itself. We had fewer B heterozygous, and this is typical in legumes, where most genotypes will be inbred. So this is a measure of how inbred the mung beans are. We had only 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 individuals, with each dot representing an individual out of the 95 that were not homozygous for either, in this case, the allele T and T from nucleotides and DNA, or C and C, the blue, for the alternative allele. That's just a close-up, and the close-up of the 96-well plate. When we looked at population structure with this, we found that basically we could divide into two groups, but it wasn't all that meaningful because the population structure is not as definitive as you would find it in maybe some other legumes that were bred over and developed and domesticated over a wider region of the earth. But we did find a little separation of East Asian and South Asian genotypes. And especially the Southwest Asian seem to have diverged from the rest of the Asian material. And then material from Asia has gone to Africa, mainly from South Asia, which would be explicable by people going from one place to another. But from East Asia, less germplasm has moved to other countries. So moving to a mineral analysis, which is part of biofortification, we have an ICP machine in our in an adjacent laboratory, and these are called inductively coupled plasma OES machines because they can detect a basic set of minerals, and these minerals range from boron, calcium, so how much calcium is in a seed, all the way to iron and zinc being two important minerals for people in developing countries where these deficiencies are quite extreme. But it's also an interesting point for consumer acceptance here in the U.S., especially since mung bean are often associated with vegetarian diets, whether those be people of South Asian descent or vegetarians that want to go vegan or want to go replace meat. Mung bean actually is used as a plant protein source and is found in your egg substitutes, so your vegan eggs. The results of the mineral analysis with those 95 genotypes and the 400 genotypes, depending on the year, 2016 and 2017, showed a normal distribution for most of the minerals. And unfortunately, some low correlation between years, but correlations within year that were significant between minerals that we normally find associated, like iron and zinc. Okay, so iron and zinc normally are somewhat associated around R.4, and sure enough, that was the case. But when you move from soil to soil across different years, sometimes you'll have some variability in the seed minerals. So that's something to consider when breeding. I just wanted to show the phenotyping side of being in the field. This is an aerial picture of our field in the middle of summer where we've plowed down a crop of winter rotation, and we've grown our mung bean, and it's surrounded by a corn border. And here there's 400 little plots representing the entire core collection that we did at the beginning to have mung bean as a source for mineral analysis, which is shown on the right. Hey, Tristan, are people seeing my cursor?
Yep, we can see it.
Great. Okay, so I'm glad I'm using it, not for any reason. So the next step in genotyping was to actually do genotyping by sequencing, which again is a 96-well plate with a little bit more difficult flow through to get information that is bioinformatically deconvoluted to find SNPs. So again, SNPs come out of this diversity analysis based on sequencing. And this was really interesting because it sort of confirmed our CASP analysis, which was done with genes. And we found grouping, especially where anything that was from Iran, Afghanistan, this drier environment where maybe it's grown, with very little supplemental irrigation, is different from the material that comes from South Asia, India, also East Asia being a little bit separate up at the top. South Korea is represented in the middle. You see Philippines and Thailand. And one of the few materials for the U.S. was actually in the group from this dry area rather than from this wetter area. So this principal component analysis, coordinate analysis, really gave us sort of a idea of what we're working with. We're working with a species that has somewhat high diversity, and it can be maybe diverse along a transect of germplasm from areas in South Asia all the way to East Asia. When we did genome-wide association tests, and these are Manhattan plots across the 11 chromosomes of mung bean, we did find certain associations for SNP markers with iron, that's here in red, and with zinc, that's here in purple. And you can see there's a greater number of genes involved with zinc. That tends to be more variable across soils than there is with iron. When we looked at iron, we had a total, in this case now, of 6,400 SNPs, and the Manhattan plots with iron and zinc association showed 43 of these SNPs associated explained 22% of the overall variation in seed mineral contents. So that's all of them, calcium. But 38 of these genes were interesting from a point of view of blasting to find out which might be most important. So in this case, we had used 14 minerals in the analysis, and in the future, we plan to do 26 minerals with the same strategy of ICP. And we have a new U.S.A. project where we can do some of this work with a greater number of minerals. Some of these are interesting from the point of view of heavy metal accumulation, and some are interesting from the point of view of, nutraceuticals in the U.S. You can see on the heavy metal side, cadmium and lead. And I think that a lot of these crops will be grown in more organic systems down in the valleys where soil is richer, but you also have a problem with some industrial contamination. We certainly do in our site along the Cumberland River in what would be the general Tennessee Valley Authority area. So a summary of results was from the GBS, was a GBS marker analysis, uncovered 6,000 SNPs useful for diversity analysis. Their prior CASP assay development successfully converted 41 sequences into 20 new and robust CASP assays. Of these, 18 markers were polymorphic with a total of 38 alleles. polymorphism information content, etc., more of specific interest. Results 2 for population structure analysis grouped 90% of the accessions into 2 subpopulations with 10% accessions belonging to an in-between or admixture group. And this model-based clustering doesn't follow many geographic origins of germplasm, but did suggest a Southeast versus South Asia divide in the case of GBS. AMOVA analysis and principal coordinate analysis sort of tell you the fidelity of these analyses. So the discussion is that this was the first time in the U.S. to attempt a large-scale SNP polymorphism in mung bean. The screening of germplasm collection is part of mung bean breeding, and this requires that cultivar diversity be fully characterized to identify potential parents without genotypic bias or without selecting material that are too close to really provide any meaningful genetic diversity in crosses. In this study, we investigated the molecular fingerprints of 95 mung bean genotypes from the USDA core collection for the species using the newly developed CASPs and the GBS technique. And the samples represented 27 countries across 10 regions that we thought would be useful to introducing mung bean to the U.S. The genes targeted were important in terms of proteins they produce and could be useful in panel markers for further association study analysis. And I just wanted to point out a mineral uptake and transport function gene called yellow stripe. This is a gene that has been found to be really important for iron uptake. A second gene that we found important was a purple acid phosphatase, and either of these could be interesting for how much iron is detectable in the collection. So we have sort of a gene target and a genetic or germplasm group target for our breeding work. So in conclusion for this part, and I'll go into one little practical area that is a current master's student project, we have SNP markers that are useful for future germplasm characterization, and some of these are the first designed for 3-primer system of CASP detection. And we hope to select for some of the genes, including this YSL one for iron micronutrient content. Now I'll go into some selections that we made. We have a variety from our very initial work in 2015 that you saw around the borders of the plots. And we've done some population development from a mixed seed source. And this has been done over the period 2016 until COVID. And then we did some seed multiplication during COVID, but we got hit by some hurricanes and realized that one aspect of mung bean that's a little bit difficult is to get too much rain at the end of the season. So that's a limitation for some areas of the Southeast. We wanted to compare to work that's being done in Alabama at the 1890 University, Alabama A&M, and also in Oklahoma, where there's a certified seed program for 2 varieties from a breeding program in the late 1990s that developed OK 2000 and Brecken. One is a mung bean for sprouts, and one is a mung bean for protein. And they're both very heat tolerant, but we wanted to see how they did in a wetter climate. We also wanted to see how trials followed wheat as a commercial crop or cover crop in till and no-till systems, because this is something that's become really popular in Tennessee, is to have wheat before soybean, mainly because of weed problems, but also because of input costs. And lately, because of the war in Ukraine, where wheat is increasing in price. And this is where I've recruited a student under a new USDA and FFAR project. His name is Josh Relier, and he's doing a master's on these basically all green types. We're not so sure where their genotypic background is from in terms of the material from Oklahoma and Alabama, but they're probably a mix of Asian genotypes and still slightly unpurified, so they have some of this darkening of seed. And mung bean has many beneficial crop traits. It's fast growing. I think I didn't mention that it can be ready in 3 months. It's versatile in a range of climates, and it can be grown either in the end of the spring season or right in the middle of summer, because it does so well as a seedling, and then it can grow into the fall. So it could be planted maybe either twice a year or in 2 different flanks of the summer season. And this is mainly because mung bean mimics the adaptation for areas of, at least for Tennessee, for germplasm from areas of China. So the origin of mung bean is in this part of the world, in China, where the blue arrow is pointing, and the red hardiness zone indicates that it's a long summer season. And basically, you can see that germplasm can move from China to the southeast, where there's another area of red climate hardiness zone. And this spreads all the way to Texas, Oklahoma, Arkansas, Louisiana, and within the southeast. The thing we've got to look at is where areas of too much rain might affect mung bean. The materials and methods that Josh is using in the field is field plots within the wheat, that the wheat's been harvested, and then he's put in these 3 varieties of mung bean. 4 varieties of mung bean is the plan with the TSU, but we didn't have enough seed this year. So Alabama A&M variety, Brecken, and OK 2000. The growth is quite luxurious. It makes a great cover crop. It has few diseases and insects, but we're scouting for them. And then at the end of the season, you can see that when we keep the area around where the wheat was mowed, we have a good crop of mung bean. And we're doing 3 experimental sites in the FAR project. One is in Nashville, one is in Memphis, and one is in a little town called Carthage, where we're on a commercial and a non-governmental organic CSA farm, which is actually the farm that Al Gore developed for capturing carbon, so carbon underground. And we're concentrating our results so far on Nashville, Tennessee, because this is all from this year. And so we've harvested from Nashville, Tennessee. The Tennessee State University Nashville experiment included tractor preparation of 4 plots for early June, and those were 4 repetitions with 3 varieties, Alabama A&M, Brecken, and OK 2000. The watering was a little shaky this year because we had such a severe drought, but we had 96 rows with 8 per variety within these plots. So split plot design. We did hand weeding and hand harvesting in September and October, and half the plots were for following commercial wheat, and half were not following a good crop of wheat. And we managed it with small tractors, as you would on a small organic farm. You know, most of the organic producers in Tennessee, which actually has the highest number of farmer markets in the country, or one of them, people don't have big machinery, but actually mung bean does really well with lightweight, small John Deeres like this one. This is just initial results in Nashville with 2022 yield data. We're measuring in grams per 10 meter of row, and you can see that no-till versus till conventional wheat preparation makes a difference in terms of growth of the plants, and that certain varieties have an interaction with that. So the 2 Oklahoma varieties have a similar result where they yield lower, about 400 grams per 10 meters in no-till, but following wheat, they do better. And this is partly because of the we think, the good soil prep that the wheat does over the winter. So the main reasons for mung bean production, to wrap up, in Tennessee are to solve a lack of plant protein sources. I mentioned the vegan eggs. One company is called Just Eggs. To diversify legumes for southeast of the U.S. and to introduce mung beans into the American diet. So we're working with some food scientists and consumer scientists to develop an outreach strategy for mung bean and help with health issues around the world. This is one of the really good, available area sources of iron. And I'd like to acknowledge Josh for his slides. He's the MS student right now. We have a postdoc, Dr. Lee, and I'd like to acknowledge her. Our previous master's student is now at Texas A&M, and his name's Fareed. He was from Bangladesh and very familiar with the crop. So was Shimbo Wu, who's currently a UF professor. And then we had visiting scholars from Malaysia and a research associate from Kenya. And then we'd like to thank Brad Morris from the U.S.A. Germplasm Bank at Griffin, Georgia, and the funding from Borlaug, FAR, and USDA. So with that, I'll open it to any questions, and thanks a lot for your attention. I already see some chats.
All right. Thank you so much. It was a wonderful presentation. My name is Nathan Johnson. Everybody listening, I'll be kind of moderating some of these questions. So if you have questions, please put them into the question and answer box. So Dr. Blair, first, so this is kind of a going organic type of seminar. And so I want to start with kind of hopefully it's a little bit of a softball question, easier. We'll see. So how would you adapt mung bean, or you can talk about other crops you work with, to organic agriculture? Where do you begin? What hurdles would be unique to mung bean?
That's a good question, and I'm sorry I didn't, more specifically address that in this talk, because I knew I was going into an organic environment, but I see people like Diego Rubiales and other germplasm people, so I wanted to address their interests too. The organic in Tennessee State is run at two sites that are a little bit off the site that I use. So I am not using an organic environment for selection, even though we've produced that work that you saw from Josh, the MS student. He used absolutely no pesticides, not even any inorganic fertilizers, not even any inoculum. So we were trying to isolate to see if natural rhizobia infection was occurring for nitrogen fixation from organic sources with this mung bean as a cover crop and as a new grain source. But I think that the selection in organic environments is important. And so following what Dr. Dill does in Clemson and at the PD station, it's really great to work with farmers directly. And we've gotten a sense of that by going over to this small town, Carthage, and planting under big plots. So we gave them about 4 pounds each of seed and said, go ahead and plant this as you would. And they planted it in their organic CSA vegetable section that they weren't using for one summer and that had rotated out of watermelon, I believe, in the previous year, and that had barley as a winter crop, and barley and vetch, I believe. Josh can correct me if I'm wrong. If you put into those organic systems, just like what we're trying to do with the wheat rotation, we threw some vetch into our winter wheat. And we didn't talk about that because the vetch came up a little bit, in different places. So we didn't really consider it a wheat vetch field. But we did try to go with a low input system. So I think that's very familiar to people that are under organic agriculture. So I hope that answers the question a little bit in terms of how a nice new legume like this can fit in. There are some limitations. There's no certified organic seed that I know of. But then organic producers allow you to use non-organic seed if there's nothing else available. So those are some of the limitations of working with a new crop. Yeah, definitely. And so the hurdle of working with a new crop and then also working strictly organic for me is not feasible right now in Nashville, but we are definitely doing it in that site in Carthage. Now, the Memphis site is actually, we didn't mention that, but it's the other extreme. The AgriCenter farm that we're working with actually is the testing site for all herbicide combinations for the southern region. They even have a herbicide judging contest. So they're the opposite of organic.
I see. Thank you for that. So here's a question in the chat. What does mung bean taste like?
Oh, great question. So mung bean has a very neutral flavor. It's the least beany of the beans that I've ever tasted. And I wish somebody would analyze that really well. But it basically can be used almost directly as a flour, which it is in India, where it's mixed in with pancakes. You have mung bean pancakes. You have mung bean directly into dal. And then obviously in Thai culture, the bean noodles are made from mung bean. So if anyone's tasted mung bean noodle in a Thai restaurant, if they're from the U.S., you probably have done that as a really exciting thing to do. And so you know what mung bean tastes like. It's basically a nice starchy taste. And you're getting all the protein as an extra bang for your buck.
Nice. Which leads to the next question. What is the range of seed protein concentrations among the accessions you have tested?
Oh, that's a great question. We have some data, I think, from the USDA, and we haven't done as wide testing as we want. So I'll hesitate to say for our samples, but from the literature, it can go up to 25% protein. And so it's a little bit less than soybean, but you're not dealing with any of the oils or the heavy dose of phytoestrogens that you have in soybean. So I'd say that the protein is a good balance. And it's similar to common bean, which I saw a little blurb on for comparison to meat. And it was amazing how well some legumes compare to meat in terms of iron and some other things. Availability is another issue. So some plant proteins have a little bit more of an issue with being readily digestible. And most of the proteins digestible is mainly a problem you have with the starch.
So it's the starch that's hard to digest? Is that what you're saying?
Yeah. Usually the starch is the least digestible. I think that mung bean, if it was better studied, would show that it's very low in resistant starches. And so this is what makes it difficult to digest to a certain extent, but also some of those resistant starches are the fibers that are healthy for you. So that's one thing. And then there are these complex sugars that are the main factor that we always talk about with beans, which is the musical fruit.
Oh, yeah.
The more you eat, the more you chew.
Yeah. All right. So kind of looking over at the chat here, I had a question about soil type for mung bean, when to plant it. Could you speak to some of those general questions about mung bean?
Oh, sure. Yeah. I see some good questions in the chat, especially in Mississippi and other areas. So the soil type for mung bean is, I'd say, a wide range of soil types. So they tend to be on the more neutral pH soils. They often follow rice in their normal systems in Asia. Especially in subtropical areas where they can grow well into the dry season just on the remnant moisture from rice. It'd be perfect in Arkansas and Louisiana, places that grow rice, to try that, to see if there's long enough season. It can be planted up until July. We've been able to get a crop, but that's in Tennessee, so you could probably plant it even late July further south. And then you can plant as early as the soil warms up. So you could plant in mid-May. And our experience has been on richer soils. So we've planted in Memphis, which is typical Mississippi alluvial soils, right in Shelby County. So it's a well-known experimental site and farm. It's the main farmers market area for the city of Memphis. And then Carthage, which is river bottom, just like our two sites where we planted before. And so we get quite luxurious growth. And I think the Oklahoma, Texas growers have a little bit sandier, harder clay soils, and they still do well out there.
Great. All right. So we're about out of time, but nice question here and the question answer. It is not unusual that a new crop suffers little diseases and pests. However, they used to increase when the crop get established. Which are the most critical diseases, and how would you cope with them in organic agriculture if no resistant varieties are in place?
Oh, that's a great question. Yeah. So we have seen almost no diseases in the field. And we've lucked out on some of our summers being hot and dry. So that's not conducive for diseases. But in the greenhouse, we've seen root rots. And so I believe that root rots would probably be the first problem, especially in organics where it's difficult to coat the seed. I would recommend using a biocontrol application and also rotating quite effectively. But then that's the purpose of this kind of crop. You want to rotate it. You don't want to grow soybean after soybean after soybean, and you don't want to do the same with mung bean either. So I imagine nematodes would also become an issue. And then there'll be some aerial fungi and insects that would be a problem later on. And I see one last question about commercial growers direct harvesting like soybean. It could be direct harvested. And so we have recently acquired a combine that's used in soybean, and we're going to use it in mung bean, but it requires planting on a certain distance of row width. And one thing we're testing with the group in Memphis is whether this mung bean could be planted down to 7.5 inches, and then it would need a different type of header for combining. And so we have combined in the past with SRES type experimental combine, and it does work as long as it has the real head on it. And so that just gives you an idea, but that will be a limitation for the small organic farmer. But then I have nothing against just taking a thresher out in the field and windrowing it and passing it through the mechanical thresher or electric thresher or whatever you want to use. We get really clean seed that way. The one thing that needs work is a little bit the sieving of waste products. And also there's a weed that is of a similar size seed. It's called the sweet potato vine. So wherever you have sweet potato vines, you want to be extra careful about clean seed.
All right. Well, it's 10:02. So I think I'll wrap up question and answer. Thank you for your presentation and all the great answers. And I'm going to hand it over to Tristan to finish things up.
Thank you.
All right. Well, thank you everyone for sticking with us. So our next seminar in December 7th at 10 a.m. East Coast time is Dr. Diego Rubiales. He's a plant breeder with the Institute for Sustainable Agriculture in Cordoba, Spain. So tune in with us next week or next month. Thank you so much for joining. Have a great day.
Tristan, thank you so much for hosting.
Oh, yeah. thank you for joining us. It was a great talk.
You take care and say hi to Dill. Say hi to everybody. Say hi to Kresovich. He's a friend of mine.
Oh, yep, will do.
Okay.
Thanks again. We'll be in touch.
Bye.